Hello, everyone, and welcome to today's live broadcast, Genome in a Bottle, Developing Benchmark Sets for Structural Variants, presented by Justin Zook, Research Scientist, NIST. I am Alexis Corrales of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Zook. I will now turn the presentation over to him.
Invitation to talk about the work we've been doing as part of our Genome in a Bottle Consortium. And I'll give a little bit of background information about the work we've been doing in Genome in a Bottle for all types of variation in a genome. And then we'll focus specifically on the work we've been doing developing benchmark call sets for structural variants, which are a more challenging type of variation in the genome to assess. So the main things that I'll be talking about today that I'd like you to get out of this is that Genome in a Bottle's mission is to authoritatively characterize human genomes. And our current characterization of the genome enables benchmarking of easier variants and regions, like small variants and mostly non-repetitive regions in germline genomes. And this has really been used widely in clinical validation and in technology development and optimization and demonstration. But it still leaves out a lot of the more challenging variants and regions of the genome. And so we are now working on more difficult variants and difficult regions. And in particular, I'm going to focus on our work on larger variants today. So we've made some draft variant calls greater than 20 base pairs in size available and have been collecting feedback about those calls and are continuing to collect feedback. And now we're working on finalizing a tiered benchmark set greater than or equal to 50 base pairs that would also have high confidence regions that could be used. There are lots of challenges in doing this and lots of work to do in getting to more difficult variants and regions. And this is an open science project and collaborations are very welcome. So please let me know if you're interested in participating in our work. So first, why we're doing this work as part of Genome in a Bottle and why we formed the Genome in a Bottle Consortium. So the need is really that, to help to know whether the answer that you get from sequencing a human genome is correct. And of course, when you use genome sequencing in diagnostics or precision medicine, this really requires high levels of confidence. And so it's important to have benchmarks that can be used. And you'll see this this Venn diagram on the right-hand side from several years ago that compared a lot of different small variant callers on an exome sequencing data set, and only about 60% of the variant calls agreed between those 5 different variant callers, even when using the same underlying sequencing data. And of course, this has improved over time. And for the easier regions of the genome, we really get quite high concordance between the different methods now, especially for small variants. But once you get into more difficult variants and more difficult regions, there's still a lot of discordance between different methods. And so the approach we've been taking to address this challenge of different methods disagreeing with each other is to develop some well-characterized genomes and just a small number of them to benchmark performance of sequencing methods. At NIST, one of the things we do is create reference materials, which are really well-characterized materials. In this case, it's extracted human genome DNA from cell lines, and we have 5 genomes that we've made available as reference materials from NIST. These are also available as both DNA and as cell lines from the Coriell cell line repository. And we've sequenced these with many different methods and tried to disseminate benchmark variant calls that can be used and benchmark regions that can be used to evaluate your sequencing method's performance. And we've also done a lot of work towards providing benchmarking methods so that you can compare your calls to ours in a standardized way and make sure that you're getting out accurate and meaningful performance metrics in the end that you can compare across experiments. So first, talking about sort of what is Genome in a Bottle, we are, as I mentioned, we're doing authoritative characterization of human genomes. So we're not trying to characterize lots of different samples, like it's not a population study. This is really a small number of genomes that we're trying to sequence as deeply and with as many different orthogonal methods as we can. We created the first human genome reference materials that were characterized on a genome scale. We have 5 of them that we've released now. We have an enduring commitment to resource availability, and this includes both enduring commitment to make these samples available really broadly, as well as to making the data available. And this is both the raw data, the analyses that are done on those data, as well as the high confidence benchmark calls that we make available. We really have put a lot of emphasis on making these widely available open resources so that there are no restrictions on their use or on distribution. That means that there have been a number of commercial products that actually use these samples as a basis for their reference samples that are aimed at more clinical uses. And there are no restrictions on publication on these data, and so we really encourage anyone to use these data as part of their methods development process, because that then helps us also in characterizing these genomes. We really try to enable technology development and tool building by disseminating these benchmark samples, and this has been used in development of new technologies, in optimization of new technologies and bioinformatics methods, and then to demonstrate the performance of these methods when they're released. The germline samples that we've been developing are available now, and we'll likely continue to develop some additional germline samples. And then we're also developing capacity for cancer samples or somatic samples, but this is a longer term work, and we don't yet have any samples that are available for that. That said, there has been some work that's been done on mixing together our germline samples to sort of simulate somatic mutations, and that can be useful as one of the things that you do as part of understanding the performance of somatic variant calling. So Genome in a Bottle has sort of evolved from when it first started in 2012, where really there were no human benchmark calls available. So we formed this Genome in a Bottle Consortium. This consortium includes people developing sequencing technologies, people from other government institutes, and a lot of bioinformatics developers, both at academic labs as well as companies, and also people developing reference samples and clinical laboratories. In 2014, we released our first, we published our first small variant genotypes, which covered about 77% of the genome in our pilot genome, which is the well-known sample NA12878 from Coriell. In 2015, we released that as a human genome reference material from NIST, which was the first reference material characterized on a genome scale. In 2016, we characterized 4 more genomes from the Personal Genome Project, and we released new small variant calls that covered now about 90% of these 5 genomes. And it also, we made calls for both GRCH37 and GRCH38, so you could benchmark on either of these references. And then for the past couple years and going forward, we're really focusing on characterizing difficult variants and difficult regions of the genome for these germline genomes, as well as developing tumor samples and then developing methods to characterize these tumor samples. So we've characterized 5 human genomes so far that we've released as NIST reference materials. The pilot genome is this NA12878 sample. And then the other genomes that we've characterized are from the Personal Genome Project. And we've done this because it enables commercial products to be developed from them. And they're really broadly consented so that there are a lot of different uses that can be applied to these genomes, and you could, and I'll have a slide about this later about some of the different uses that people have already had for them, and there are many others that we hope that people will come up with in the future. So we have an Ashkenazi Jewish trio, that's a mother, father, son trio, and a son in a Chinese trio where we are also characterizing the parents in the trio, but they are not released as reference materials, that they're only available from Coriell. We've chosen to work with, to characterize trios like this because that can help us in both understanding the accuracy of our variant calls by seeing whether variants and genotypes are inherited as we'd expect them to be. And it also helps with things like phasing, and some methods can even take advantage of this trio information to better characterize the genomes. So we have released all of our data openly as quickly as we can, and we've also tried to describe these data as well as we can so that anyone can use these. One of the ways that we described data in the data that we collected up through 2016 is through a publication in the Nature Journal Scientific Data, which allows you to publish data sets without doing extensive analyses on them yet. And so this allows anyone in the community to know what data is available and how they can use these data as part of their pipelines. This was a really big effort that included many different authors and institutions. There were 12 different types of data that were published in this, from short reads to linked reads to long reads to optical mapping methods. And it was data from 7 different genomes, and the data were described in detail so that anyone can use these. We've made these data openly available and all of our analyses openly available because this has resulted in sort of a virtual open science. So the cycle that sort of we continually go through is that users can analyze the genome in a bottle samples, either getting the DNA or cells and sequencing with a new method, or taking the data we've already made available and analyzing that data with new methods. And they can use that for method development, for optimization and demonstration by benchmarking the results that they get versus our genome in a bottle benchmark data sets. This could be part of their assay validation process if, for example, it's a clinical laboratory that's doing this. And then the other thing that we ask everyone to do is give us feedback about the calls that we've made available so that we know whether there are any weaknesses in our current benchmark data sets that we could help to address by incorporating in the new methods that people are developing. So if it looks like any of these new methods that are developed are going to be useful for improving our high confidence calls or for expanding to more difficult regions and more difficult variants, we'll integrate those new methods into our integration process, release new benchmark data sets, and then users can again compare to these, and we'll go through the cycle again over and over again to iteratively improve. And this is really important in genome sequencing because the technologies and the methods are evolving so rapidly. And so it's important for us to basically make sure that our benchmark data sets stay ahead of these new methods or keep up with the new methods so that anyone can benchmark the new methods and know how well they're doing for more difficult regions and for more difficult variants in the genome. The other thing this open science enables is secondary reference samples. So there are now more than 30 products that are available from several different institutions that are based on these broadly consented, well-characterized genome in a bottle cell lines from the Personal Genome Project. So we've been really grateful to work with the Personal Genome Project in this and grateful to the individuals contributing these samples to the Personal Genome Project because they enable a lot of innovative products. And these products so far have included things like the genomic DNA from our cell lines plus synthetic DNA spike-ins. And these synthetic DNA fragments might have clinical variants in them of interest, since our cell lines don't necessarily have particular clinically interesting variants in them. They've also had variants spiked in at different allele frequencies to mimic somatic variation. And then there's also a product that we've helped to characterize that has difficult variants in it. So these are difficult variants in clinically targeted regions so that clinical laboratories can assess how well they're characterizing these more difficult variants in clinically interesting regions. And this is really important because the variants in our cell lines tend to be mostly SNPs in clinically targeted regions. And so if you're doing a targeted panel, you also really need to have other types of more difficult variants that you can understand your performance of. And this type of sample helps with that. There's also work been done to put these cells in a matrix that would mimic what clinical samples would look like. So for example, in formalin-fixed paraffin-embedded samples, there are some that are being developed to mimic circulating tumor DNA. There are now stem cells available from Coriell, and there may be more of these developed in the future. And then in the future, there are some groups that are looking at editing these genomes where you might be able to edit in particular variants of interest for benchmarking. So the approach that we've taken to develop high confidence variant calls for our genomes is to integrate a diverse array of data types and a diverse set of different analysis methods to characterize our genomes. And so we have a lot of data that we've made publicly available, and we've both characterized these data ourselves, but a lot of the work has been done by the community, the Genome in a Bottle community, to apply new methods to all of these data that we've made available. So we have deep short read sequencing, linked read sequencing like that from 10x Genomics, long reads like those from PacBio and from Oxford Nanopore, and then optical and nanopore mapping data like that from BioNanogenomics and NAPSIS. And then we have a lot of different analysis methods. So we have released benchmark calls based on small variant calling methods, and we're now working on structural variant calling and also incorporating in results from local and global assembly methods. And this is another sort of cycle that we go through where we try to discover and refine sequence resolved calls from multiple data sets and analyses. By sequence resolved here, I mean that we think we know what the actual sequence change is, as opposed to sort of just knowing that roughly some region of the genome might be duplicated or deleted. We compare the different methods variant calls to each other and their genotype calls to each other. We evaluate those calls using all of the different data sets that we have. We try to identify features associated with the reliability of the calls from each of the methods. And these might be things like mapping quality for that particular data set at that location, or strand bias, or base quality scores, or clipping of the reads, or a number of these different features that are often output by variant callers. And then we use these features to understand when we can trust each method. And so if they disagree with each other, we trust ones that sort of look like good normal calls, and we form our benchmark calls using mostly heuristics up to this point with a little bit of machine learning, and we're exploring more sophisticated machine learning methods to integrate all of these data together. And then to evaluate the benchmark calls that we get, we compare these to other high quality call sets that you might want to benchmark against these calls, and then manually curate any differences between other call sets in our benchmark. And our goal in the end is to result in calls where if you compare to our benchmark calls, the majority of putative false positives and false negatives are actually errors in your data set rather than errors in our benchmarking call set or errors in like the comparison process. So our small variant calls have undergone an evolution from what we published back in 2014, which was version 2.19, to the calls that we published last year in version 3.3.2, where initially we'd covered about 77% of the genome. Now we cover about 90% of the genome. The number of variant calls has increased pretty substantially, and the number of indels has also increased substantially over time. One of the ways we've evaluated our calls was by comparing to platinum genomes, which I've abbreviated PG in these slides. And the number of differences between these call sets has decreased since our initial call sets. And when we go in and actually look at these differences, we find that a pretty small number of the differences are actually errors in the NIST high confidence calls. And so we really roughly estimate that maybe there are about 2 false positives and 2 false negatives per million NIST variants that are inside our high confidence regions and also inside the platinum genomes high confidence regions. And we can't really assess the performance of our calls outside the platinum genomes high confidence regions. So it's a little hard to know exactly what the error rates are, but we talk about this in some more detail, including some evaluations we did based on trio Mendelian inheritance in this preprint that's linked to at the bottom left of this slide. The other thing we've added to our calls over time is phasing information, and this is mostly based on using the pedigree that NA12878 is part of, or the trios that the other samples are a part of. And we haven't done a lot of evaluations of these phasing yet, so we're now working on trying to incorporate some of the newer phasing methods and using things like linked reads and other new phasing methods to add additional phasing information and additional confidence to that phasing. The other thing that's been really important is to develop benchmarking tools that anyone can use to compare to are high confidence calls. And so you might think that this is sort of a straightforward thing to do, where you just like take your variant call list and compare it to our variant call list, and you can just say how many are the same and how many are different, and that gives you the performance metrics as an output. But there are a lot of complications when you get into the details of this. And one of the initial things that we did as part of this Global Alliance Genomics and Health benchmarking team is develop standardized definitions for performance metrics like true positive and false negative. And the way we chose to do this is to define different levels of stringency when you're doing the matching. And so in some applications, you might want to make sure that both your variant calls and your genotype calls are correct. And then we only count it as a true positive if both of those are correct. And that's actually the default matching that we use with our tools. But we also output other statistics that allow you to see how many of those errors are genotyping errors, or how many of those are places where there is actually a variant in that region and you're just calling the wrong variant in that region. The other thing we've developed as part of this team is sophisticated benchmarking toolkit that sort of is integrated into a single framework that has standardized inputs and outputs. And there were a number of different sophisticated benchmarking and variant comparison tools that were developed to handle cases like the complex variant that's delineated on the right-hand side here. And so one of the challenges when doing comparisons is that variants can be represented in different ways. And some of the hardest cases are cases like these complex variants, where you have a set of bases that's replaced by a different set of bases, and that could be represented just as a single variant like it is on the top, or it can be represented as, in this case, deletion and a SNP as it is on the bottom on the right-hand side. And there would actually be even more ways of representing this variant, and there's not really a clear way to standardize representation in all of these cases or to normalize representation in all of these cases. Normalization can help, but it tends to not resolve all of the different challenging representation cases that occur in the genome, because a lot of these occur in repetitive regions, and there are enough of these complex variants in the genome that they end up contributing pretty significantly to your false positives and false negatives if you don't use a sophisticated benchmarking tool. Then finally, what we've done is develop standardized bed files that allow you to stratify your performance by genome context. And so it allows you to see how well you do for your variants in easy, non-repetitive regions of the genome versus different types of repetitive regions of the genome, whether these are different lengths of homopolymers or different lengths of tandem repeats, or difficult to map regions of the genome. And this allows you to know sort of where you might need to improve your methods, or where the weaknesses in your methods are. And we've made these tools available on GitHub. They're also described in a lot of detail and with some examples in a preprint that's linked to here in the bottom left-hand side of the slide. The other thing we've done is worked with the Precision FDA team to make these tools available in the Precision FDA interface. And this is a nice web-based interface to our benchmarking tools that allows you to simply upload your variant call file, select which genome it's from, and so which benchmark VCF and bed file you want to compare to, and then we'll output the performance metrics in a standardized way, and it will output all these stratifications that I've mentioned. And this was also, our tools and our benchmark call sets were also used as part of a couple challenges that the Precision FDA hosted, where the community could contribute analyses of data from our genomes, and so anyone can see sort of how these methods compare to our call sets and how they perform in these different genome contexts. And you can go to the Precision FDA site, and anyone can apply for an account there and view these results, as well as do your own comparisons and either make them public or not as you choose. So we've done a lot of work on small variants, but there's still a lot of the genome that's challenging to address. And I've at least sort of roughly tried to estimate sort of what we're still missing in the genome. And this is not a like really precise quantitative estimate of what we're missing, but just to give a sense, for simple SNPs and non-repetitive regions, we're probably calling about 97% of the variants, and in our high confidence, and we're covering about 90% of them, 7% of them in our high confidence regions. So we might have about 100,000 or so variants that we're still missing in this region, and then there are also simple indels in non-repetitive regions and we're maybe missing about 7% of those. But then we're missing a much larger percentage of variants once you get to low mappability regions, once you get to and of course regions that aren't even in GRCH37 or GRCH38, we're not characterizing at all at this point. There are small indels that are in tandem repeats and homopolymers that we're not yet accessing. We're characterizing probably less than 50% of these right now, and so there are many more that we have yet to characterize. And then as you get to larger indels and structural variants, we're missing many of these in our current characterization. And so a lot of our work right at the moment is focused on these. We have also started work on difficult to map regions and tandem repeats and homopolymers. We're interested in extending our approach to the approaches that we've used so far to structural variants, and there are a lot of similarities to what we've done for small variants, where we can collect call sets from multiple different technologies and multiple different calling methods. We can compare these call sets to each other to find calls that are supported by multiple technologies. But there are a lot of challenges that we've had to address that are different from small variants. So for small variants, call sets tend to have pretty high sensitivity to variants if you set the parameters correctly, at least in defined regions of the genome that aren't difficult to map or repetitive. But for structural variants, that's not always the case yet. Variants are also often imprecisely characterized, so there might not be exact breakpoints or the exact size or type or sequence that's predicted for insertions. The representation of these variants is poorly standardized, and that's especially true once you get to more complex changes that occur in repetitive regions. And then tools to do the comparisons of structural variants are really in their infancy, and so there have been there's been a lot of work that's been done in this in the past couple years, but we have yet to sort of come to a consensus on what the best approach is to do this yet. So our integration strategy for structural variants has been to collect many different candidate calls for our Ashkenazi Jewish trio from the Personal Genome Project. We've collected our calls from many different technologies, from short reads and linked reads and long reads, and from optical and nanopore-based mapping methods, and then from many different calling approaches, including small variant callers, structural variant callers, and methods that use local or global de novo assemblies. So we got over 1 million different calls from over 30 different methods and over 5 different technologies. Then, of course, many of these were either exactly the same or similar to each other. And so the next step was to sort of refine and evaluate and genotype these candidate calls. So one of the first things we did was ask for sequence resolved calls as often as we could, meaning we want to sort of get as close to exact breakpoints and as close to the exact sequence that's inserted or deleted as we can. And this allows us to use sort of a uniform method to integrate these call sets together, and allows us to output calls that are easier to use as a benchmark set since we've more precisely defined what exactly we think the sequence change is. And so this is often done using assembly-based approaches, either local or global assembly-based approaches. Once we have these sequence resolved calls, we compare these sequence predictions of the candidate calls and merge calls that are sufficiently similar to each other. And then we go back and if the calls look like they have enough support that we want to evaluate them further, we determine the support in the raw data for each of these sequence resolved calls, and we try to genotype them based on that data. And we've been using a tool called SVViz to do that genotyping. So we've gone through multiple iterations of integrated calls using sort of an evolving set of methods where initially we were just using deletions and we didn't have sequence resolved calls. Then we moved to using sequence resolved calls in version 0.3, and we did clustering based on that. In version 0.4, we used SVViz to evaluate the calls as well as using the optical mapping and nanopore-based mapping methods. We also added genotypes for all three members of the trio in this case. And then in our current latest call set, version 0.5, we used some new calling methods that were developed, especially to help to call larger insertions in a sequence resolved way. And we also included more calls that were only called by a single technology because we found that some of these looked like they were likely to be true, but we evaluated them with the raw data to make sure that they were supported. And we added some preliminary phasing information, though this is still a work in progress. And the work that we're currently undergoing and will continue to work on in the future is to resolve some cases where we have clusters of differing calls, because we find that, so in some cases, these are actually, you have two different calls on the two different alleles, or you might have two calls that are nearby each other on the same allele. But there are a lot of other cases where there's one correct call and one incorrect call at the same location, and we're still trying to work on ways to resolve these cases. One of the ways we're working on that is by improving phasing information that we have. We're also adding in new data types and improving the sequence resolution of the calls. And then we're also working on developing high confidence regions so that you could look at both false positive and false negative rates for your calls. The set of evaluation and genotyping methods that we've used up to this point has been primarily using SVViz, which maps reads to the reference and alternate alleles that are hypothesized and sees which allele the reads support better. And we've done this with PacBio reads. We've also phased the PacBio reads using 10x and a tool called WhatsApp, so that we could see whether the support for heterozygous calls segregates by allele or by haplotype. And we've also used Illumina paired end and mate pair data, as well as 10x data that's been haplotype separated. And then we've also compared the sizes that BioNano predicts and that NABSIS predicts to our calls to enable us to assess the accuracy of larger insertions and deletions. In the future, we're continuing to work on better separating haplotypes for our data sets, doing that and adding in additional phasing information from the trio as well. We're working on manual curation of these calls, and I'll have a slide later about with a link to a beta version of this. And starting on around May 8th or so at the Biology of Genomes conference, we'll hopefully have a large set of calls that's made available, and it'd be great to have anyone participate in that curation. We're also working on methods for doing targeted sequencing of these calls to evaluate them using, for example, PCR amplification and then Sanger sequencing. And if anyone is interested in helping with this, we're also really open to collaborations for that. We, just as some preliminary analyses for version 0.5, we've made some size histograms where we show smaller calls over here between 50 and 1,000 base pairs on the left-hand side for deletions and insertions, and then larger calls on the right-hand side up to 10,000 base pairs, though we do have calls that are larger than that also. And on the left-hand side, you can see a nice peak for ALU insertions around and deletions around 300 base pairs in size. And on the right-hand side, you can see a nice peak around 6,000 base pairs for line insertions and deletions. So this at least helps us to have some confidence that we're calling some real things that we'd expect biologically. Moving forward beyond the version 0.5 call set, one of the things we realized as the Genome in a Bottle community tried to compare to these version 0.5 calls is that it would probably be useful to work on a two-tiered system of calls where the tier 1 calls would be simple calls that are isolated where we've been able to sequence resolve them. So these would be an insertion or a deletion that's not near any other insertions or deletions that are large, and where we've been able to sequence resolve them. So we think we know approximately what the sequence change is. And so this is probably going to build on our version 0.5 call set. We're probably going to focus on calls greater than 49 base pairs in size, even though we've made calls smaller than that. And focusing initially on HG002, which is the son in our Ashkenazi trio. And so we sort of used a set of heuristics to say that they had to have sufficient support from different technologies or multiple call sets, and that they needed to be supported by SVViz analyses or by the optical or nanopore-based mapping methods. They also, in this case, we excluded a lot of calls that were within 1,000 base pairs of another greater than 49 base pair call. And it turns out that something like a third of the calls were within 1,000 base pairs of another putative call that looked like it was well supported. So this excluded a significant number, but now we're down to about 14,000 calls that we think are isolated so that you can use them in a better way for benchmarking your call sets. But we didn't want to stick with only this tier 1 call set because it excludes a lot of these more difficult variants that we have been working on characterizing. And it's often useful to know whether you are missing these calls where maybe we're pretty confident that there is a structural variant, but it's complex, where we have multiple clustered calls, or where we couldn't come up with a consensus sequence change at that location. And so in this case, we merged calls without requiring that their sequences were similar to each other, and are coming up with a set of, in this case, about 2,600 regions where there were clustered calls. And then we also clustered calls that were not tested so that they weren't supported by two or more technologies or 4 or more call sets in our initial clustering. And found that still there were about 9,000 regions where multiple technologies or many call sets supported the call in that region. They just didn't agree on what that call should be. So this tier 2 call set would be useful for maybe assessing your sensitivity to more difficult calls, but we wouldn't be able to test sort of how accurate your call is, or maybe or whether you're calling the exact rate change at that location. Where the other thing we wanted to do in addition to developing this 2-tiered call set is develop a high confidence bed file that allows you to assess false positive rates. And so to do this, we have been working on a method where we're using assemblies to develop this bed file. So we first are calling variants from each assembly and exclude regions around any well-supported variants from these assemblies that are not in our version 0.5 call set. And then we find regions for each assembly that are covered by one contig if it's a haploid assembly, or by two contigs if it's a diploid assembly. And we're still working on developing the heuristics that we'll need to set thresholds of sort of how much support do we need to make this a high confidence region where we're pretty sure that we're calling all of the variants in that region. So we're hoping to have this have an initial version of this here in the next few weeks, and if you're interested in sort of engaging in this, you can sign up for our Genome in a Bottle analysis team email list, and I'll have a link to that at the end of this presentation. So in addition to developing heuristics and integration approaches, we also wanted to develop ways for us to manually curate these sites, because we found that often it's really useful to actually look at the raw data in these regions to understand the support for the variants. So a postdoc in our group at NIST, Leslie Chapman, has been working on these, developing a web-based manual curation tool that allows you to see visualizations of data like that from IGV, from PacBio and 10X and Illumina and other data sets. And then also you can view the results from SVViz and dot plots for this region. And we'd like to collect input feedback from as many people as possible in the community about whether people think we're calling the correct variant at this location, what the genotype of the variant is, and how much confidence you have in that, or whether this is a more complex region. And we're in beta testing now, and so you can go to the link in the bottom left and you can sort of play around with it now, but we're not yet really collecting feedback other than feedback about the actual interface to help us improve it. But we're planning to launch this around May 8th or so very soon, and volunteers are really needed to help us to manually curate these sites, and encourage you to participate in this. So there are a number of challenges that remain, including getting to larger sequence resolved insertions. We've made a lot of progress on this, but we're so that we now have more similar numbers between large insertions and deletions, but still have some work to do for larger insertions. There are a lot of these dense or clustered calls that are different from each other, where we still have to figure out how to resolve them, and we need to resolve places where we might have an inaccurate sequence change or an incomplete sequence change. We're working on developing bed files to define homozygous reference regions. And then we're also working on benchmarking tool development, because there's a lot of need to understand how you compare your variant calls to ours, and then understanding what performance metrics are important for different applications. There's a new tool that's really just been made public by Spiral Genetics that's still like in beta testing at this point, but it looks like it incorporates a lot of the work that we've been doing in Genome in a Bottle so far in different ways of comparing structural variants, and encourage you to take a look at this if you're interested. Also wanted to let you know that we're developing new data sets. We have new data from 10X Genomics available. We're developing PacBio data from the SQL instrument on the Chinese trio, as well as additional data on the Ashkenazi trio that's currently undergoing QC, and so should be made available very soon. We'll have new bionano data from their DLS labeling method available very soon. There's a new linked read method from Complete Genomics that we're making available soon. And then we're also in collaboration with University of Birmingham and Nottingham in the UK, we're working on developing ultra long reads from Oxford Nanopore. We're preliminarily getting really long reads, like 80 to 90 kb and 50, probably at lower coverage across the trio than our other data sets, but still likely to be really useful for resolving some of these complex regions. And then we're also looking into potentially getting strand-seq data, which can help with phasing and inversion detection. And then we're also looking at new samples, and this includes samples from additional ancestries, as well as developing cancer samples that we'll characterize in the future. So the road ahead is sort of characterizing large variants and difficult small variants and phasing in the next year and probably going forward. Then in 2019, characterizing more difficult larger variants and even more difficult small variants, as well as starting with somatic sample development and new germline samples. And then longer term, looking at things like diploid assembly and how do you use those as part of our variant calling process, as well as how do you benchmark them. Looking at somatic variation, segmental duplications, and centromere and telomere, that there's a lot of work to do in characterizing difficult regions of the genome yet. So just wanted you to sort of take home that we've been working on authoritatively characterizing these human genomes, and really encourage you to use our data and use our analyses of these data and give us feedback on them. We currently allow you to benchmark easier regions and are actively working on more difficult regions. We're also actively working on making new data sets available, and so if you're developing methods, it would be great to have you try out your methods on these and share your analyses with us. So we hope that these will help with clinical applications and enabling precision medicine to reach its promise, as well as many research applications in understanding how new methods are measuring difficult variants and regions. So I'd like to acknowledge that this is the work of a large number of people, both at NIST, as well as in the Genome in a Bottle Consortium and the Global Alliance for Genomics and Health Benchmarking team, as well as support and collaboration from the FDA. So finally, if you're interested in additional information, there are a number of links here on this last slide, and happy to receive any questions or feedback by email or as part of this webinar as well. And our next public workshop is scheduled for January 2019, tentatively, probably at Stanford University in California. So thanks much for your attention, and I'm happy to take any questions now.
Thank you, Dr. Zook, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window, type your question into the box that appears on the screen, and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, beyond structural variants, what are other challenging regions and variant types that Genome in a Bottle plans to characterize?
Yeah, so we've currently been focusing a lot of our work on structural variation and trying to develop a benchmark set for that. But we have started a number of discussions with groups that are looking at things like characterizing variants in difficult to map regions using linked reads or using long read information and using phasing information. And so, so there are a number of methods under active development from several different groups that show a lot of promise in characterizing these regions. And so we're working on how we can with these groups on how we can integrate the results from those methods into our high confidence calls, so that anyone can benchmark their calls in these difficult to map regions. And sort of similarly, we're looking at integrating phasing information using some of these same methods, and then also calls in tandem repeats and homopolymers, where right now we're not yet fully characterizing these, and a number of people have been working on developing methods targeted to these regions that we hope to incorporate within the next couple years.
Our next question is, can anyone participate in the manual curation of SVs for Genome in a Bottle?
Yeah, so we definitely are happy to have anyone participate in this, regardless of your expertise level. And so we plan to have some sort of training that you can go through, or some example sites that you can look through to understand how best you can understand the different types of visualizations that we give in this web-based app. And it'll be great to have anyone. And of course, it's great to have experts participating in this if you already have a lot of experience looking at these structural variants. But we hope that but in some initial work that we did, it looked like even if you're not an expert, you're likely able to give us useful feedback about many of these sites to help us to understand their accuracy.
Dr. Zook, how do you expect new methods using long and ultra-long reads to be useful in defining benchmark calls?
Yeah, so we've definitely already been finding these to be really useful in characterizing structural variation. And so in particular, when we're trying to characterize this structural variation in long repetitive regions, or in regions, or in places where there are large insertions, it's been really useful to have long read information, and we're really excited about getting the even longer reads that we're working on, because we expect this will help us to continue to get characterizing longer repeats and longer insertions with more confidence. And then these are also really useful for phasing variants, and it looks like they're going to be really useful for characterizing variants in difficult to map regions as well. Though that's sort of an ongoing process, since these reads do have a higher error rate in general, and so characterizing small variants with them is taking a substantial amount of methods development to use them in that way.
And it looks like we have time for one more question. Do you have plans to use new graph genome methods, and do you foresee any challenges with benchmarking these methods?
Yeah, so we've definitely been talking with the people who are developing these methods using graph genomes. And so for those who might not be familiar with them, there are methods being developed where you can incorporate known variation in the population into a graph-based reference. So you don't just have a linear reference anymore, but it's now a graph that has multiple potential paths in it for the different variants that have been found previously. And then you align the reads to this graph so that it particularly can help in regions where there are larger variants, or where there's a lot of diversity in the human population. So it can help with characterizing structural variation. And it can also help with characterizing regions like the MHC, where there's a lot of diversity and different people's haplotypes are quite different from the reference. And so we're excited about using these methods as part of our integration process in the future, as these methods continue to be developed. And then the other challenge related to benchmarking these methods is that potentially you could imagine the graph incorporating all of the variants that we found in our samples into the graph. And so when you're benchmarking versus our samples and use these variants in your graph, you're sort of incorporating prior knowledge about our samples into your analysis. And so it's possible that these methods might perform better on our genome in a bottle samples than they would on some new sample that has new variants that haven't yet been discovered. And so one of the things the people developing these graph genomes are really thinking about as they develop these is how you benchmark them in a way such that you don't bias your results. And so it might include like making sure they don't include all the variants that are found in our samples in their graph, or doing some other types of analyses. But I imagine that methods like these graph genomes, as well as methods that use like machine learning, like the deep learning based methods that have recently been developed, that there needs to be a special care taken to not overtrain to our samples that we're already making available. And hopefully, like having additional samples like we've been making available will help with doing that better.
I would like to once again thank Dr. Zook for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.